Hello guys, welcome back to my channel. In this video, we are going to discuss a complex topic, but I have made it very easy for you guys, and that is called as intercellular communications, or it is also called as signal transduction. Now, first of all, what do we understand by the word signal transduction or intercellular communications? It is nothing but that one cell is communicating with another cell. Now, how is it that one cell is going to communicate with another cell? Now, let's say this is the first cell and this is the second cell, and this cell wants to communicate with this cell. So what this cell is going to do is, it is going to release some chemical messengers, and these chemical messengers are going to be recognized by the second cell because the second cell has got receptors for the recognition of these chemical messengers. Now, this is how the signal from one cell is transmitted or transferred to the second cell, and in the second cell we can see some physiological action. Usually, these chemical messengers, they are secreted either into the extracellular fluid or they are secreted into the blood. But there is one exception wherein the chemical messengers are going to get transferred from one cell to another cell without entering into the extracellular fluid, and this is going to occur because of the presence of what is called as the gap junctions. Now, we all know what do we understand or what is the meaning of this gap junction. The gap junction is the one which is going to bridge the gap between the two cells. That is, there is going to be a small connection which is going to be formed between the two cells. Now, because of these connections, it becomes very easy for the cell to transfer the substances from this cell to the another cell. And gap junctions are predominantly present in the myocardium, because of which the electrical impulses which are passing between the myocardial cells occur very fastly, and therefore the myocardium is going to contract as a single unit. Okay, so one example wherein the signal transduction is occurring without the chemical messengers entering into the extracellular fluid is gap junctions, and where gap junctions are present in the myocardium, because of which the electrical transmission of impulses occurs very fastly. Apart from that, majority of the other modes of signal transductions are going to occur when these chemical messengers are going to enter either into the extracellular fluid or into the blood, and they are going to go and bind to the receptors which are present in other cells. So where these receptors could be present, these receptors could be present on the cell membrane of the other cell, they could be present in the cytoplasm, or they could be also present on the nucleus. Next, let's understand as to what are the different types of communications which are present. The first one is what is called as neural communication. So the classical example what we can give for neural communication is the synapse. So we all know what is the synapse. Synapse is a junction between the two neurons, and this neuron is what is called as the presynaptic neuron, and this neuron is what is called as a postsynaptic neuron. How do you think the signal transduction occurs here? The presynaptic neuron is going to release these chemical messengers which are called as the neurotransmitters, and these neurotransmitters are going to go and bind to the receptors which are present on the postsynaptic neuron. And the best example of the neurotransmitter which we can give here is acetylcholine. Second type of communication is wherein one cell is secreting the chemical messengers, and these chemical messengers are traveling in the bloodstream throughout the body, and then they go and act on the target cell, whichever target cell is having the receptor. Whenever a chemical messenger is entering into the bloodstream and communication is occurring by this means, that is what is called as endocrinal mode of communication. Next is what is called as the paracrine mode of communication. Paracrine mode is nothing but one cell is secreting the chemical messenger and the target cell is present very nearby to the primary cell. This is what is called as the paracrine. So it could be a neighboring cell or it could be in the vicinity. So here the chemical messenger is going and acting on this target cell. The best example, this is what is called as a paracrine mode of communication. The best example I could give you for paracrine mode of communication is the histamine, which is secreted from cells which are called as the ECL cells. What are these ECL cells? These are nothing but enterochromaffin like cells, and these cells are going to act on the parietal cells. So where are ECL cells and parietal cells located? Both are located in the gastric glands. Okay, so it is very much nearby. So the histamine produced from the ECL cell is going to go and act on the parietal cells, and then the parietal cells are going to increase the secretion of hydrochloric acid. Next mode of communication is wherein a cell secretes a chemical messenger, and this chemical messenger is going to act on the same cell. Such a kind of communication is what is called as autocrine communication, and the best example is insulin, which is secreted from the beta cells, acts on the beta cells itself. Now let's understand two important terminologies with respect to the receptors. One is called as down regulation and one is called as the up regulation. Now what is the meaning of down regulation? Down regulation means decrease in the number of receptors. When do you think the number of receptors is going to decrease? When the hormone or the chemical messenger which is secreted is increased in amount. So whenever there is an increase in the secretion or increase in the rate of secretion of the hormone, the number of receptors are going to reduce. That is called as the down regulation. And what is up regulation? Up regulation is entirely reverse of that. Up regulation is increase in the number of receptors. Why? When the hormone which is secreted is less in amount. Very easy. Down regulation and up regulation. Next, let's understand the most important part of this video session, that is the mechanism of communication. First mode of communication is what is occurring through change in the membrane permeability. So this is a diagram wherein this is our cell membrane, and in the cell membrane we are seeing these receptors. What receptors are these? These are ion channel receptors, and these ion channel receptors are having one extracellular part. One more is what is called as the transmembrane part, another is the intracellular part. So the extracellular part of these receptors, it is having the binding site for the ligand. Okay, here the ligand is nothing but a chemical messenger. So once this ligand or the chemical messenger comes and binds to these sites, these ion channels are initially in a closed state. So once there is binding of these ligands to that extracellular portion of these receptors, which are nothing but the ion channel receptors, what is going to happen here? What is happening here? What we are seeing here is that these ion channels are opening up. So once these ion channels open up, what is happening in this example is that the sodium which is there on the outside, now it is going to enter inside the cell. So what has happened is the conductance of the cell membrane has increased. So the membrane permeability has increased, and these receptors are called as ion channel linked receptors. Again, the best example I can give you is acetylcholine, which is acting on the nicotinic receptors which are present in the neuromuscular junction. So this is one mode of communication wherein the action is occurring through change in the membrane permeability via the ion channel linked receptors. These ion channel linked receptors are closed. They are having an extracellular domain to which the ligand is coming and binding. Once the ligand comes and binds, or a chemical messenger comes and binds to the extracellular domain, the ion channels are going to open up. So opening of the ion channels is going to cause movement of the sodium or any ion from outside to inside, and then there is going to be a response in that cell. This is one mode. Second mode is action which is occurring through the effect on the gene expression. So here what is happening is, this is a hormone and this is a binding protein. So this hormone along with the binding protein, actually this hormone is transported using the binding protein to the cell. So once it comes near the cell, what is going to happen is this hormone is going to move or diffuse across this cell membrane. So one very important property of this hormone should be that this hormone should be lipophilic hormone. This hormone should be a lipophilic hormone. That means it should have the ability to dissolve in the lipid bilayer. So once it has dissolved in the lipid bilayer, the hormone has now entered inside the cell. So once it is inside the cell, what is going to happen is it is going to bind to the receptor which are present in the cytoplasm. So these receptors are called as intracytoplasmic receptors. Sometimes the receptors can be also present in the nucleus. So now what is happening is a combination of this hormone and the receptor is going to form, it's going to form a complex. This is called as the hormone receptor complex, or it is also called as the ligand receptor complex. And now this complex is going to enter into the nucleus. So once this complex has entered inside the nucleus, it is going to stimulate a part of the nucleus which is called as HRE. What does HRE mean here? HRE means hormone responsive elements. So once there is stimulation of the hormone responsive element, there is also stimulation of the process of transcription, because of which there is production of the mRNA. Now this mRNA which is produced inside the nucleus, it is going to come out of the nucleus and it is going to stimulate the Golgi apparatus, and the Golgi apparatus is going to increase the production of certain proteins, because of which there will be a physiological action in this cell. Got this? So this is called as action which is occurring through the effect of gene expression. First and foremost thing which is required for this is that the hormone should be a lipophilic hormone. I will give you the examples in the last slide. Until and unless it is lipophilic, it is unable to cross the lipid bilayer and enter inside the cell. Next mode of action is a very important mode of action. Here the action is occurring through special group of receptors which are called as G protein coupled receptors. What these are called? G protein coupled receptors. So once there is stimulation of this G protein coupled receptors, there is also production of chemicals which are called as the second messengers. So let's understand the mechanism or how the structure of the G protein coupled receptor and the mechanism as to how these G protein coupled receptors are stimulated. Again, we can see here that this is our cell membrane, and in the cell membrane we are having this receptor. Now this receptor has got an extracellular domain, it has got a transmembrane domain, and it has got an intracellular domain. The extracellular domain of the receptor is the one which is allowing the ligand or the hormone to bind to it. The transmembrane domain is very important because the transmembrane domain is traversing the cell membrane 7 times. You can count it. This is 1, this is 2, 3, 4, 5, 6, 7. That is why this receptor is also called as a 7 transmembrane receptor, or it is also called as serpentine receptor because it looks like a snake. Now coming to the intracellular portion of this receptor, the intracellular portion is the one which is attached to a protein which is called as a G protein. That's why this receptor is called as a G protein coupled receptor. Now what is this G protein? G protein is having a trimeric subunit. What are the 3 subunits? One is an alpha subunit, second one is a beta subunit, and the third one is the gamma subunit. Now attached to the alpha subunit is the GDP. That is why this protein is called as a G protein. Now, once the hormone comes and binds to this extracellular domain of this receptor, what is going to happen is the GDP, which was attached to the alpha subunit, is going to get converted into GTP. So once the GDP has got converted into GTP, we say that the G protein has become active. So once there is activation of the G protein, what is going to happen here is that the alpha subunit is going to dissociate from the beta and the gamma subunit, and this alpha subunit is going to go and activate membrane bound target proteins. What are these called as? These are called as a membrane bound target proteins. That means these proteins are also present in the membrane, and these membrane bound target proteins can be of 2 types. They could be enzymes which are present in the membrane, or they could be also ion channels. And if they are enzymes, they are going to further help in the production of the second messengers, because of which the physiological actions in the cell is going to take place. Okay, this is the mechanism of G protein coupled receptor. Now what happens once the hormone is removed from the receptor? Once the hormone is removed from the receptor, the GTP which was formed is going to get converted back to the GDP. Now why does this occur? Because the alpha subunit has got an intrinsic GTPase activity. So now once the GTP gets converted back to the GDP, the alpha subunit which had dissociated from the beta and the gamma subunit, now it goes back to the gamma and the beta subunit. Okay, remember that the alpha subunit, what we are talking about, it can be either stimulatory or it can be inhibitory. If it is stimulatory, we write it as alpha S. If it is inhibitory, we write it as alpha I. What are the enzymes which can be present in the membrane which could be stimulated? The first enzyme is called as adenyl cyclase. And because of the stimulation of adenyl cyclase, this generation of cyclic AMP, this is called as the second messenger. There could be stimulation of the guanyl cyclase. Why these enzymes are getting stimulated? Because of the activation of the G protein. The G protein which has got activated, the alpha subunit along with the GTP, it is going and stimulating these enzyme systems which are present again in the membrane. So these are also transmembrane enzymes like adenyl cyclase. So once adenyl cyclase has got stimulated, it is going to generate cyclic AMP. Cyclic AMP here is the second messenger. Similarly, guanyl cyclase is stimulated, it is going to generate cyclic GMP. There could be also stimulation of what is called as phospholipase. This is called as membrane phospholipid phospholipase system. So once that is stimulated, there is generation of 3 second messengers. These are called as inositol triphosphate, diacylglycerol, and also calcium. So these are the 3 enzymes, and there could be also stimulation of the calcium channels, and stimulation of the calcium channels is going to cause binding of calcium to intracellular chemical substance which is called as calmodulin. So this system is called as calcium calmodulin system. So let me just give you an example of how does this adenyl cyclase cyclic AMP system is going to act. Now again, this is our cell membrane, and this receptor what we are seeing here is the G protein coupled receptor. It is having an extracellular domain, intracellular domain, and a transmembrane domain. So to this extracellular domain, let's say there is binding of the epinephrine. So once the epinephrine binds to the extracellular domain, what I said is going to happen, the GDP which is attached to the alpha subunit, it is going to get converted into the GTP. Once that occurs, the alpha subunit is going to dissociate from the beta and the gamma subunit, and then it is going to go and activate these transmembrane enzymes. Now in this case, the transmembrane enzyme is called as adenylyl cyclase, or it is also called as adenylate cyclase. So once there is activation of this adenylate cyclase is going to cause conversion of ATP into cyclic AMP. Now this cyclic AMP is going to cause stimulation of protein kinases. So protein kinases are going to get stimulated, and there is going to be the response of the cell. So what is this cyclic AMP here? The cyclic AMP is acting as the second messenger. So if the cyclic AMP is the second messenger, what is the first messenger? The first messenger is the epinephrine. Similarly, there could be also stimulation of guanyl cyclase resulting in the formation of cyclic GMP. Now in that case, cyclic GMP is the second messenger. Next, let's understand how this membrane phospholipid phospholipase system is going to act. Again, this is our G protein. This is the peptide hormone which is coming and binding to the G protein. So there is now activation of the G protein. Now activation of the G protein is going to cause further activation of one more membrane enzyme which is called as phospholipase C. So once there is activation of the phospholipase C, this phospholipase C is going to cause breaking down of PIP2. So what is this PIP2? Phosphatidyl inositol 3,4 diphosphate. Now it is going to break down into two compounds. One is called as inositol triphosphate, another one is called as diacylglycerol. So both IP3 and DAG are what? They are our second messengers. They are our second messengers, and the peptide hormone is our first messenger. So once the IP3 is formed, this IP3 is going to dissociate into the endoplasmic reticulum, and it is going to cause stimulation or increase in the release of the calcium, because of which the cell's response is going to act. Hence, even our calcium is going to be called as the second messenger. Now what the diacylglycerol is going to do is, it is going to stay in the membrane itself, and in the membrane itself it is going to cause activation of protein kinases. So once there is activation of the protein kinases, this results in phosphorylation of the proteins, and hence there is going to be a cell response. So second messengers are our cyclic AMP, cyclic GMP, inositol triphosphate, diacylglycerol, and the calcium. Next, let's understand what is the meaning of this calcium calmodulin system. So here also the receptor comes and binds to the G protein. So once the G protein is activated, this is going to cause opening of the calcium channels which are present in the membrane. So once there is opening of the calcium channels, there is going to be a calcium influx. So once the calcium concentration inside the cell increases, this increase in the calcium concentration inside the cell, it is going to cause stimulation of the endoplasmic reticulum, and endoplasmic reticulum is further going to increase the concentration of the calcium. This is called as calcium induced calcium release. Now the calcium which is present inside the cell in a very high concentration, it is going to bind to one protein which is called as calmodulin. So this combination of calcium and calmodulin, it is going to either increase or decrease the enzyme activity, because of which there is going to be an increase or decrease in the metabolic pathways. Now one very important enzyme which is activated by the combination of calcium and calmodulin is what is called as calmodulin dependent protein kinases, and in that the most important is this enzyme which is called as myosin light chain kinase, which is extremely important for the contraction of the smooth muscle. Coming to the last mechanism, this is the action which is occurring through tyrosine kinase activation. So what is special about this receptor here is that this is a receptor which is spanning the cell membrane only once, unlike the G protein which spanned the cell membrane how many number of times? It spanned the cell membrane 7 times. And this receptor, the intracellular domain of this receptor has got the enzyme in it. Okay, intracellular domain only is having the enzyme. So what is going to happen is, once the hormone comes and binds to the receptor, the intracellular domain of the receptor has got autophosphorylation site. So once there is autophosphorylation of these sites, the intracellular domain of this receptor is going to become tyrosine kinase. It itself is going to get converted into tyrosine kinase. That is what is called as primary substrate phosphorylation. So once there is primary substrate phosphorylation, further there is going to be phosphorylation of serine and threonine kinases, because of which there could be gene transcription, or there could be also enzyme activation like protein kinase activation, and hence there could be physiological effect. Sometimes what happens is that the intracellular domain of this receptor may not have tyrosine kinase, but the tyrosine kinase can be present separately inside the cytoplasm. Such kind of tyrosine kinases are what are called as Janus kinases. So even at that point of time, if the hormone is coming and binding to the receptor, these tyrosine kinases which are present in the cytoplasm, they are going to become active. And what are they called? They are called as Janus kinases. And these Janus kinases are going to activate signal transducers and activators of transcription proteins. Hence, this pathway is what is called as JAK-STAT pathway. And we should remember one very important hormone which acts via this pathway, that is our growth hormone. So to summarize as to how is it that the signal transduction is going to take place, I told you that signal transduction, for it to occur, there should be release of chemical messengers, and usually these chemical messengers have to enter either into the extracellular fluid or into the blood. But there is one exception wherein the chemical messenger is not going to enter into the extracellular fluid, and that is through the gap junction. Next is these chemical messengers can cause signal transduction through a change in the membrane permeability. Very good example is our neuromuscular junction and the synapse. How does that occur? That occurs via the ion channels. Next is, there are hormones which can cross the cell membrane and they can go and form a complex with the receptors which are present either in the cytoplasm or in the nucleus. And this mode is what is called as through the gene expression because of the intracellular receptors. Next very important topic we have studied is there is signal transduction occurring through what is called as G protein coupled receptors and the generation of the second messengers. And at last we have studied how the signal transduction occurs through tyrosine kinase activation. So these are very important. These are the one which are asked in your examinations. Via membrane permeability, example is acetylcholine. Directly via gene expression, these are the lipophilic hormones. These are the thyroid hormones, all your steroid hormones. So when I say steroid hormones, I include sex steroids like estrogen and testosterone. I include corticosteroids like cortisol. I include mineralocorticoids like aldosterone. Okay, and also vitamin D. To this list, you can also add vitamin A, that is the retinoids. The one which are acting via the G protein coupled receptors and via the activation of adenyl cyclase and hence via the generation of cyclic AMP are norepinephrine and GnRH. Okay, that is gonadotropin releasing hormone. The one which are acting via activation of the membrane phospholipase C is angiotensin 2 and antidiuretic hormone. The ones which are acting via activation of the guanyl cyclase, all these are via G protein coupled receptors only. And hence via the generation of the cyclic GMP are two more hormones. One is called as atrial natriuretic peptide, another one is the nitric oxide. And the one which are acting via the tyrosine kinase mechanism are the insulin and the platelet derived growth factor. I know this topic is little bit difficult. Listen to this video once again. If you have any queries, leave them in the comment section below. I will try my level best to solve them. Thanks a lot.